like to do now is return to the schedule and introduce Ana Maria Cuervo, who will speak to us about selective autophagy, or fighting aging one protein at a time. Ana Maria is co-director of the Einstein Institute for Aging Research. She is a member of the Einstein Liver Research Center and Cancer Center, a professor of medicine at Albert Einstein College. Her work focuses on lysosome protein degradation, which is autophagy. And this is in association with neurodegenerative diseases like Parkinson, Alzheimer, Huntington disease. She's received an awful lot of awards for her superb work, and I presume is also a clinician. So, Dr. Cuervo, I'd like to hear from you.
Thank you very much. I know that it's getting late on the day, so I will try to go brief, and hopefully we'll have some time to discuss. And I'm very glad we have in the middle some talk, because I didn't want to pass from food to garbage. That is mostly what I'm interested in. So the break was nice in the middle. But basically, why should I even care about garbage? So our main interest on garbage is actually because my main concern is in aging. So I really would like to tell you a little the point of view of our lab is that in order, or the secret to live longer and healthy, I think it's all about cleaning. And probably I inherited that from my mom. She was very much into cleaning. But I really think that this is the case, because when you think about all these age-related disorders, I mean, this is something that occurs in aging. So the way that we go about the problem is, if we can slow down aging, we might slow down Alzheimer's or cancer or diabetes. So there is a very, very important component that aging contributes to these diseases. And from the simplistic way of a cell biologist as I am, the idea is that if you can keep your cells as when they were young and they were clean and healthy, you will prevent the accumulation of that damage and all these disorders inside the cell. And of course, the cells have a variety of cleaning tools, as we all have at home, and we are interested in some of them. And just to refresh a little your cell biology, if you remember, when you have a protein that is damaged or is altered, it can be degraded by this big chopping machine that is called the proteasome. But even the proteasome is a gorgeous protease and a structure in the cell. The problem is that it can only degrade proteins. But this is not the only thing you have in the cell, and this is not the only thing that gets damaged every day and every moment. So in order to deal with all these other structures, you have an organelle that is called the lysosome. And you all probably remember it as the garbage container of the cell when you study cell biology. But the beauty, as I say, is that it not only degrades proteins when they are damaged or when they aggregate, but it can also degrade organelles. So you remember your mitochondria, your endoplasmic reticulum, any other structure that has to be continuously renewing the cell, it can be eliminated in this lysosome. And also any pathogen, anything that came from outside and released in the cell, it can be eliminated through these lysosomes. And this process of degradation of your own material is what is known as autophagy, kind of eating yourself. But I would say more than eating, should be recycling yourself, because you are utilizing the components of all these structures, the amino acids of the proteins, the lipids of the membranes of these mitochondria, to recycle them and utilize them for new synthesis. So I would say that the ultimate recycling compartment in the cell is really this lysosome. So we've been very interested in this idea of autophagy and sending things to lysosomes for degrade. And I'm not going to go into the details, but just because it will be important in the talk, that there are different ways in which you can take something from the cell and deliver it to the lysosomes. You can either wrap it in vesicles, and this is a process that is called macroautophagy, because it will mostly take organelles and big things in the cell. but you can also take single proteins and deliver them one by one across this membrane. And of course, you can already see the benefit. This will have very high capability, this macroautophagy, and this other process that is mediated by chaperones, like the surveillance systems in the cell that determine that this green protein is something bad that you have to eliminate. This other process is much more selective. So it will not be able to take a whole organelle, or it will not be able to take a whole compartment, but it will be able to discriminate protein A from protein B. And if this is the damage, only take the damaged one and leave the other healthy and around. And this process, as I mentioned, is called chaperone-mediated autophagy. So the lysosome itself and the process of autophagy was known since before even I was born, since the early 60s. And it's also the discovery of the tube. And in the last, I would say, 10 to 15 years, there has been a whole revival of this pathway, of this structure in the cell. And this is what got us very excited. And the revival is mostly because the components that participate in each of these steps, the genes that encode for the proteins that are going to make those vesicles or that are going to make those transporters, now they have been identified. So then you can start eliminating them and see what happens when you cannot clean, when you cannot eliminate these products, or you can overexpress it and then see what is the beneficial effect of having more cleaning or returning to the normal cleaning in a cell that has no longer been able to do that. And thanks to all these studies, and this is really the whole field of autophagy, we have learned that autophagy participates in many functions that are essential or very important for the cell. So the first one, and this is how it was described, is energy. And this comes because of the idea of recycling. So for example, if we just have lunch, we didn't give you a break, so you didn't snack in the middle. So before dinner, your liver, for example, is going to activate this process of autophagy. And it's not because it has to clean. It's mostly because it needs nutrients, and you are not providing them because we have you here, sit down and close, with no food. So basically your cells, your liver is going to activate this to recycle materials that might not be so necessary in order to sustain the function of these cells. And of course, if you prolong the starvation, not that we are going to do that to you, but then you will activate this process of autophagy in a much more selective manner. At the beginning, you can go in bulk, and then you will become much more selective. But energy and recycling of materials is really extremely important for the cell, and it's in large part done by these lysosomes. The other function is the one that I mentioned, quality control. Anything that is produced in your cell has to be right, and anything that is damaged needs to be eliminated. So that goes without saying. But then, of course, it has other functions. I talk about how you can eliminate pathogens, bacteria, virus that came into the cell. So it's a function of defense. This system has an important role. Also in remodeling, when you think from an embryo to a normal baby, how many things have to happen, all this remodeling of tissues in part requires the activity of the system that has the proteases and the hydrolases that are going to degrade the materials around. And this also allows for reprogramming. You can change completely the composition of your proteome, of the proteins in your cells, just by activating this pathway. So these are just a couple of examples of functions that have been attributed to this process of autophagy. So we know that as you get old, one of the systems that decrease in the cell is your capacity to perform autophagy. So it goes without saying that as autophagy decreases, as you get old, you're going to have compromise in each of those functions. And this become interesting because a couple of years ago, last year, there was this paradigm paper in the field of aging biology, that is the field that I'm interested, about the 9 pillars of why we get old at the cellular level. And when you look at those things, DNA damage, protein damage, energy sensing, cellular senescence, all those processes now have been experimentally linked to a decrease in autophagy. Of course, there are many other reasons that contribute to each of those alterations, but a failure of autophagy, a failure of this clean machinery that we didn't pay much attention for a long time, seems to also contribute to each of them. And then, as I say, when experimentally we start eliminating autophagy and we start seeing what happened to the animal models or the different experimental models, or we start looking at patients of particular diseases and we try to see how are their cells cleaning, how is this autophagy system, then we can establish connections with many disorders. And I'm just going to flash some of the ones that have been really related to aging. So for example, neurodegenerative diseases, Alzheimer's, Parkinson, Huntington. Unfortunately, these are diseases that we know that the incidence increase tremendously as you get old. And in all the cases, has been described that these cleaning mechanisms in the neurons of these patients are not so efficient, and autophagy is compromised. Similarly, as you get old, your immune system declines. And now there is very nice evidence that the autophagy system of these immune cells, it also declines. And if you restore it, you can restore part of the immune capability of the defense systems in the cell. Similar things of cancer, metabolic disorders because the alterations in energy, or even osteoporosis. And now there are very nice studies that involve that if you enhance the autophagy system, you might delay the process of osteoporosis in the matrix of the bones. And as you see, these are disorders that are going to be affecting, or are affecting already, our elder society. So we think that acting on the autophagy system might have really something of good on that. So as I say, we are cell biologists in the lab, so we see, or we look at this in a very simplistic way. And our main interest is really trying to see how can we manipulate these systems in order to slow down aging. So for any of these pathways, and I'm not going to tell you much about many of them, but the questions are always the same in the lab. It's like, why these pathways, when these mechanisms of cleaning do not work as you get old? What are the consequences? Because I'm trying to tell, oh, these are very important pathways, but we have to really show whether or not are important and which one is more important or less. And then the most important question, can we fix it? Can we do something or manipulating the systems to enhance cleaning in the old cells, and does this has any beneficial effect? So rather than going through all the pathways, I wanted just to show this particular pathway that I mentioned is very specific, that is the chaperone-mediated autophagy, because that one is particularly close to my heart. Actually, I started working on that pathway when I was a graduate student in Spain, in the lab of Erwin Neck, and I was extremely fortunate that in the first year of my thesis, as we started working with that, there was another guy in Boston, Fred Dice, that published something very similar to what we were saying, that proteins can go into lysosomes one by one. This was not very common because people thought lysosomes just go in bulk. So this one by one doesn't make much sense for lysosomes. So we realized that we might be looking at the same thing, and both of them have really the vision of say, well, let's use Anna Maria to go back and forward of the Atlantic and try to figure out if they are the same. And these are the times that I can still travel with radiolabeled proteins in my pocket in the plane. So I was going from Spain with my radiolabeled proteins, going to Boston, doing the experiments, coming back with more radiolabeled proteins from Boston into Spain, and trying to figure out if this pathway was the same. So I had the best of both worlds, of course, because I was spending winters in Valencia, that is where I was doing my thesis, gorgeous place by the water, where my father lives. And I was spending summers in Boston, escaping the heat of Valencia and having a great time in Boston. So these arrows went back and forward within 3 years. And basically, at some point, they just went one direction, and I decided to do my postdoc with Fred Dice in Boston. And then I just moved to my independent lab, working also on the same pathway. So this particular mechanism of cleaning has been, as I say, very close to me for a long time now. And just to tell you a little how that works, so what we have learned from the cell biology. So if this is the lysosome, you remember this is the bag that has all these hydrolases, what we have learned is that the main components are going to be this green chaperone, like the chaperone in the cytosol that is going to identify what has to be eliminated. And then this other blue protein, this docking protein or lysosome associated membrane protein, that basically is going to be the docking station for whatever you want to target to the lysosome. So the process more or less goes this way. You want to eliminate the red protein, you just bring it to the lysosomes, bind to this receptor. This is a single transmembrane protein that forms now a translocation complex, the protein cross, and then it gets rapidly degraded. So basically to this cartoon that takes 2 seconds, it has taken like 15 years of my life to figure out each of the components. But of course, it's a little, we have a couple more of that. We have 8 components now. But this is basically, it has been very important to understand how to then manipulate this pathway. And during this time that we were looking at this system, one of the things that we were very interested in, how is that protein being recognized and being eliminated? And we discovered that there is a sequence, there is 5 amino acids in the proteins that go through this mechanism that seems to be present in almost 40% of the proteins in your cells, and that when they get exposed, they basically can be now saying, oh, a mesa protein, take me to the degradation system, and this is what is recognized. And something that became very interesting when I set my own lab is that this motif was present in many proteins that has been shown to accumulate in diseases or be the cause of some of these diseases of neurodegeneration. So for example, some of you might hear about this alpha-synuclein, that is the protein that accumulates in the neurons of patients with Parkinson, or tau in the case of Alzheimer or frontotemporal dementia, or Huntington, that in the patients with Huntington. So these are proteins that the mutant variants get accumulated in the form of aggregates, and that alter the normal functioning of the neuron. So something that we learned through the years is that those proteins, as you get normally, are eliminated by many different mechanisms, but one of them is this pathway. And as you get old, because the activity of this pathway decreases, now those proteins start not being able to be removed so efficiently, and this might contribute to the alteration of the homeostasis of these proteins and to the formation of these aggregates, oligomers, the toxic species inside the cell that are gonna alter functioning. So we've been trying to figure out why the activity of this pathway decreased with age, and for a long time I put all my money in the chaperone. I thought that the chaperone couldn't figure out good from bad as you get old and was not taking the right proteins. But then with the time, what we really found is that the problem was in this receptor, the docking protein that I say is in the surface of the lysosome, and is what has to grab this garbage. So for example, this is the Western blot, so the black band will be LAMP2A, and you can see in animals 3 months, 22, 3 months, 22, there is a dramatic decrease in the levels of this protein as you get old, but also look at people. So these are 25 years old, probably, so skin fibroblasts probably from people in my lab, and then the 65 years old. And this is not all. I mean, if we keep going, you can see a really dramatic decrease in almost all the cells of the body. So at least we thought, it's like, okay, at least we have a reason. We know that this protein decrease, and this is very essential for this pathway. But then, and the other important thing is that this is aging, but the levels of this particular protein or this particular receptor that you need to clean can also be modulated by other factors like diet. If we put a young animal in a McDonald's diet, basically you have the same decrease being young that I'm showing you here for an old animal. So food and alimentation has a lot to do with you getting younger or older, or at least not getting so fast. And also, unfortunately, calorie restriction will have the opposite effect. Calorie restriction, as you know, is the only intervention that has been shown to increase health span and lifespan. And we also show that it improves the cleaning systems inside your cells. So now that we know more or less why things don't work, is this important? I mean, what are the consequences of this cleaning mechanism not cleaning? And as I said, the field has done many models, and I'm just going to show you one model just to give you an idea of the consequences of not being able to clean. So we decide to take out this protein, kind of mimic aging. Let's decrease the levels of this receptor. Let's decrease the cleaning through this pathway. You have other methods of cleaning in the cell, but only eliminate this one and then see what happened. And this is really work of a very talented MD PhD student, Jamie Snyder, and a postdoctoral fellow in the lab, Jose Rodriguez. And basically what they did is to take out this SLAM2 protein, this receptor protein, take it out only in the brain, in different parts of the brain. So they create different models. We have a full body, but we also have animals that do not have the cleaning system in particular areas of the brain. And then see what happened to them. And as I say, other systems of cleaning are okay. This is the one that we took out. So when you do that, you can see that the animal, so the top one will be the normal reaction of an animal. You grab it from the tail and spread the legs. When you get these animals that cannot clean the neurons in particular areas of the brain, look what happened. The guy just stayed there, and this is called clasping. It's a typical feature of neurodegeneration. When you do models that resemble Parkinson or Alzheimer, they kind of have this phenotype. And then of course we look into these animals and we start seeing, so the top will be one of these proteins that I mentioned, alpha-synuclein, that is the protein that accumulates in Parkinson. And when you look at our animals, you can see these red balls that corresponds to this alpha-synuclein in aggregates. So basically, by just eliminating the cleaning, these animals don't have the Parkinson mutation, don't have anything related to neurodegeneration itself, but just by not letting these neurons to clean their proteins periodically, you can mimic what you see in patients with neurodegeneration. So we went a little more in detail because it's like, well, this is fine. You are accumulating these proteins because you cannot remove them. But how do you justify the different phenotypes? Why do you have this phenotype? So I'm not going to go much in detail, but we have done proteomics. We took those aggregates and the assemble and say, okay, which proteins are there and what functions are you losing in the cell that are giving you this phenotype that you just showed. So we did this proteomics, and basically you lose proteins, so these proteins will be now enclosed inside your cells, so they will not be functional. So these are very important proteins. Proteins involved in cellular defense and survival, proteins involved in cell cycle, in cytoskeleton. As you know, this is going to be extremely important in a neuron for movement of vesicles around, but also proteins involved in the regulation of the energetic metabolism, metabolism of glucose, metabolism of lipids. So sure enough, we go to the neurons of these animals, and we found that the energy levels, the ATP levels, are very low. And this probably contributes to part of this phenotype that you show of neurodegeneration. So I think by doing this kind of studies, we can start trying to understand, and we have done similar with other cleaning systems, what will be important for what, and what are the systems that you are altering. But then we know that seems to be important, and I showed you only in the brain, I can show you in all other organs, and things like cleaning is really important in all the organs. But then it's the most important challenge that we have now is, can we fix it? Okay, now we know that as you get old, and we are all unfortunately gonna get old, your cleaning systems are gonna go down. So that sounds very depressing, but is there a way back? Can we do anything about that? So you already saw this image, and this was at least one component that we know from this pathway that is very important, and that it decreases as you get old in all the cells. So what we decide is, okay, let's put this protein back in animals as they get old and see if they get better. So this is basically, as I showed you, you have your lysosomes. When you are young, you have many of these receptors, so they can really bring things inside lysosomes and break it down. Then as you get old, you have less of these receptors, so proteins cannot go inside. So now let's do a transgenic animal in which we genetically, and this was a proof of principle, I definitely don't want to do gene therapy in 80-year-old people, but just to show if this was of any relevance, or it didn't matter once you reach an age and you put back this receptor, there is nothing else to do. So we did a transgenic animal, but we didn't want to put most of this receptor when they were young. We didn't want to create a super mouse that was very strong since it was young, because what we have learned is that the decrease in the levels of this cleaning doesn't occur gradually since you are born. You are perfectly okay till middle age, at least in animals, and then is when the decline starts. So what we wanted to add is when the decline, and I'm not telling you anything new, right? But yeah, so middle age sucks. But basically, but this is the point, that it's like, okay, so let's intervene there. When your cleaning systems go down, now let's activate this extra copy that we put of this protein and see what happens. And now you're going to have to believe me on this, but we have methods to measure, for example, levels of oxidized protein. This will be a bidimensional gel, and each of the black spots correspond an oxidized protein, a protein that has been damaged. So when you are young, this is more or less what you find in the liver of these animals. When they are old, this is what you see. So you see tons of garbage accumulating there. So then the idea is that, okay, these are these guys, the ones that are in this low part. So what about the ones that have kept the cleaning for a longer time at normal levels? I mean, it could be okay, or it might just be that they're a little better. There might not be difference if this pathway is not so important because you have others. But we did the experiment, and basically this is what we saw. And of course, there was a lot of screaming going on because these guys are the same age, basically, but one of them are clean and the other not. And of course, we did functional studies. I don't have time to show you, but those livers, in this case, we were targeting to liver. Those livers were much more functional, were almost similar to when they were young. And just as a piece of hope, even if we do the intervention, once they are only 3 months before we sacrifice them before the end of life, basically they still have a much more beneficial effect and you can still restore function. So this was kind of exciting. And just as a validation that this might be probably relevant, we are very fortunate in Einstein that we have a population, and this is run by near Barzilai, of centenarians. So this is these people who really live many years. And we decide, and this is work from Caroline Park, another MSTP student in the lab, in collaboration with Fernando Macian. And basically what we decide is to look at the cells from centenarians and see how was their cleaning. So you remember the oxidized proteins, we were using it as a readout of damage. So when we look at centenarians, sure enough, they have less damage in their cells. So then Caroline said, okay, is this about this cleaning? Is it because of this pathway or just because they normally are more protected of what? So this is still a very small study, but we have been looking at people of 65 years that we already know that there is a decline. And if you compare with our centenarians, the centenarians have higher levels of this LAMP2A protein that is the same one that we put in the animals. So basically, I guess we could have gone to the centenarians and save a lot of money doing the animal, because normally the centenarians know what they are doing. So basically, this gave us also the proof that cleaning is something that probably contributes to health span and to longevity. But unfortunately, for those of us who don't come from a centenarian family, we cannot change our parents now, it's too late. So what are we gonna do about that? And that's what we are really dedicated right now. So right now we've been trying to develop molecules, compounds that can really activate these pathways back. Because as I say, gene therapy is not gonna be something that you are gonna try in an Alzheimer's patient with 85-year-old. But basically, Kishen and Antonio have been using two different approaches. We found that this particular pathway is modulated by retinoic acid receptor alpha, basically in a negative way. So we have designed like custom-made molecules that can interfere with the function of this break, of this negative effect on this pathway, and then basically get an activation of the pathway. And we are using medicinal chemistry in collaboration with Emrys Gabatiotis. And for example, the green are the first molecules that we developed. And I'm not a chemist, and I always amazed of all the modifications that they can do. So now we have some molecules at this big level. Yesterday we got the first data in vivo in animals. So we are very excited about these particular molecules. But I'm an MD too, and I know that from the time as a scientist as I am about these molecules, from now that we know that they might have something till we really can get it if they are good in the clinic, it's gonna take many, many years. And we have to go through many loops that we will do. We are very determined on that. But for more immediate application, we are using repurposing. So basically what Antonio has decided is to take libraries, and this is something that is very common these days, take libraries of all the medicines, all the compounds that are utilized in the clinic for many other reasons, for bacteria infections, for whatever you want, and then try to analyze which of those compounds, and these are NIH libraries, which of those compounds will also activate this pathway. So Antonio has done a tremendous amount of work, but basically he has identified two molecules, or two tracks, because they are used in clinic already, that seems to have a very nice activating effect on this pathway. So the advantage of this is that, as I said, they are already used in the clinic, so we will have an easy go with them. It will be easier to implement if this is the case. And of these two molecules, one of them I'm not so sure what to do yet. We are following it because it's used to kill fungi in infections, but we will use it. The other one is used in schizophrenia. And this is very interesting for us because that means that cross the blood barrier, that, as you know, is a major limitation in all these neurodegenerative disorders. So we really want to kind of follow it a little more, do more medicinal chemistry, and modify it in a way that we should be able to utilize for this point. So I think I just wanted to give you a flavor of why cleaning is important, what are we doing about that, and hopefully why this will help us to live a little longer. And then this is the current composition of the lab. This is the full composition of the lab as the years pass. And when I saw the slides this morning, I did the calculations, so we are doing okay. We are 62% human and 38% men, so we are doing okay on that. And then this is the list of all the collaborators. I mean, I'm not a neuroscientist, I'm not a metabolism person, I'm not a cancer, but I have the fortune to work with all of them because we have this common interest in aging. And of course, these are our funding agents that have kept us going. Thank you so much for your attention.